everyone. My name is Dana Howard. I'm an assistant professor in the Center for Bioethics with a joint appointment in the philosophy department. And welcome to the Center for Ethics and Human Values monthly CARE panels. CARE, the Conversation About Research Ethics program, aims to support Ohio State's shared values in the domain of research by building community around discussions of research integrity. And it's meant to really be complementary to compliance-focused trainings. We hope that these panels encourage exploratory discussions of cutting-edge issues in an evolving field. And there's nothing, I think, more evolving than the topic of discussion today on the ethics of developing therapeutics for rare diseases. So the OSU Center for Ethics and Human Values CARE program is presented with the support of the College of Medicine Center for Bioethics and the College of Public Health, as well as the Center for Clinical and Translational Sciences. And we are very grateful to have two experts who are going to be talking about the topics today. One of the most exciting promises of cell and gene therapies is their potential to tailor treatment to individuals and to improve the lives of those with rare diseases. But developing these therapeutics tends to blur the traditional line between treatment and research and invites a number of really important ethical questions that we're going to be discussing. Our external panelist is Dr. Alison Bateman-House. Dr. Bateman-House is the Assistant Professor of Population Health at NYU Grossman School of Medicine. I'm going to be using last names because we have two Alison B's, so to keep things a little bit simpler. So Dr. Bateman-House's research is primarily focused on the ethics and policy issues concerning access to investigational medical products, both inside and outside of clinical trials. And Dr. Bateman-House also serves as a co-chair of the Working Group on Compassionate Use of Pre-Approval Access, CUPA. Our OSU and nationwide panelist is Dr. Alison Bradbury, who is an assistant professor of pediatrics at OSU and is the PI in the Center for Gene Therapy at the Abigail Wexner Research Institute. Dr. Bradbury is Bradbury is a PI of the Bradbury Labs, which is dedicated to understanding disease mechanisms and developing therapeutic approaches for rare pediatric and neurological disorders. So I'm really excited about this panel. We're going to start with two presentations from Dr. Bateman-House and then Dr. Bradbury to really set the scene about the ethical issues, the opportunities, what is exciting, and also the challenges that are coming up when it comes to developing these individualized therapeutics. And then I came up with some questions, but as many of you already know, there is this sort of little Q&A tab at the bottom of your screen. Whenever a question pops in your mind, feel free to start populating those questions, and we'll definitely have time to get to a lot of audience questions at the last 20 minutes of the presentation. So thanks everyone for joining, and to no further ado, we'll let Dr. Bateman-House take the stage.
Hello, everyone. I am Alison Bateman-House. It is a great pleasure to be with you today, and I appreciate very much the invitation from Dr. Howard and her team to join you. I'm very, very glad that Dr. Howard is a doctor of philosophy, because I'm going to be raising lots of philosophical issues and not answering them. And I will just put it out on the table that I am not a philosopher. So it is very good to know we have someone who's capable in the room with us. So as mentioned, I am at the NYU Grossman School of Medicine, and I do specialize in dealing with novel medical products. In our conversation today, we're going to be talking about developing therapeutics for rare diseases. And I think both Dr. Bradbury and I are going to talk for some part of our presentations about their rare diseases in general, and then get more specialized into individualized therapeutics. So that's just to set you up that we will be sort of narrowing our focus as we go. But before I go any further, I'd like to share my disclosures with you. So Parent Project Muscular Dystrophy, the various institutes at NIH that you see listed there, Janssen Pharmaceutical and Bridge Bio, all have provided me either with research or salary support. Department of Health and Human Services, I am a member of the Secretary's Advisory Committee on Human Research Protections, so I just need to make clear that what I'm saying here are my thoughts, not the government's in any way, shape, or form. And then I wanted to point out that I am a pro bono advisor to the In Lorem Foundation, which is a charitable foundation that is making antisense oligonucleotides for individual therapeutic use available to patients. I will mention that later in the presentations. I just wanted to make that conflict available up front. So happy to talk about any of those during the Q&A period, but I think we should get into the meat of the conversation before then. So as I said earlier, there's going to be sort of this narrowing of focus. There are certain issues that come with novel therapeutics of any type. Then there's certain issues that come when you're dealing with novel therapeutics for rare diseases. They're rare disease issues. And then when you go into these individualized therapeutics, therapeutics that are intended for just one recipient, or we have this term in a few, a very small handful of recipients, the exact N of which is not necessarily specified. There are even additional issues that come in. So we're going to be talking about mainly the bottom two levels of this inverted pyramid today. But in order to really situate those two, we need to talk about the first layer, novel therapeutics, just to make sure everybody's grounded, because not necessarily everybody knows about the process of drug development. And the question that I'm going to be dealing with during the entirety of my talk is, on what grounds do we justify differentiating our treatment of these categories? Okay, so when we talk about drug development, what exactly are we talking about? So this is a slide that I've used forever and ever. It's out of date at this point, but I still think it does what it needs to do in terms of getting the point across. And note, it is from pharma, the sort of industry spokes body. So it probably is slightly skewed, but I think it is still more or less accurate for the purposes of just sort of laying out, how does a drug come to market, more or less. And this is just a regular drug, nothing special in terms of rare disease, gene therapy, or anything, just a regular drug. You can see that preclinically, before it's used in any patient, any research subject, et cetera, there is screening of numerous compounds to see if any of them have, possible activity against the target of interest. That is winnowed down into a much smaller number of drug candidates that are thought to perhaps have the activity of interest. They go into clinical trials. The phase 1 trial, as many of you will know, is frequently called a safety trial. I don't like that term because really, all through the clinical trial process, and even once the drug is released into the market, we're still learning about the safety of a product. So I like to think of a phase 1 trial as you're trying to find the appropriate dose of a drug, and you're really looking to see at what point, do I give a dose and have intolerable side effects? And if there's not a point at which, there's no point at which I am free of intolerable side effects, then that drug is abandoned, etc. Phase 1 drugs frequently are tested on healthy volunteers. But when we're dealing with things like gene therapies, which cannot be, removed from the person's body once they've received one, or very toxic drugs like chemotherapies, et cetera, they are frequently tested on patients who have the disease of interest. Phase 2 drugs is when we start looking for efficacy. Does it actually work on the disease of interest? We're still collecting safety data, but we're also looking for efficacy. And then phase 3, we might be going up against a comparator. So this is where you would hear of something like a placebo-controlled trial, where some number of people get the drug candidate of interest. Some people get randomized to either a placebo or to a standard of care drug, et cetera. I'm going through this super fast, and those of you who are familiar with this area will know sometimes we put phases together. You'll have a phase 1, 2, you'll have a phase 2, 3. Sometimes you skip phases entirely, et cetera. But just wanted to point out that, we do have this preclinical part, we have this clinical part, and all of this happens. Data is collected, in animals, in humans. A package is put together, it's submitted to the FDA, and then the FDA decides, does the totality of evidence support us approving this drug for this particular indication or not? And you can see, it takes many years, it takes many subjects in clinical trials to bring just one FDA approved drug to market. So I frequently, just to try to make people understand, how unlikely that numerical compound they heard about is going to be a cure for something, just remind them that the vast majority of drugs in clinical trials fail, either because they're too toxic to use or they're not effective on whatever the indication is. Anyway, that's drug development in a nutshell. And now that you know that, we can talk about all the other more interesting things for this particular conversation. Oops. Now, in this particular conversation, I said that we're going to be talking about, at the very end of it, these individualized therapeutics. And I just want to note that FDA says itself, hey, these individualized, in this case, ASO products, antisense oligonucleotides, are not expected to follow this traditional, 1, 2, 3 path. They acknowledge upfront, this is not possible for individualized therapeutics. And what we hear a lot of, not even for individualized therapeutics, but just for rare drugs in general, is this phrase regulatory flexibility. So if you've been listening to the news at all when it comes to ALS drugs or Alzheimer's or even COVID, you hear this phrase a lot called regulatory flexibility, and we'll be coming back to that soon. So I just want to sort of level set for a moment and say, when you're dealing with genetic conditions, and a lot of the individualized therapeutics that we're going to be talking about deal with, genetic conditions, at least at the moment. Here's the situation. Both within the United States and then, of course, globally, but I'm going to focus really within the United States for right now. There's inequitable access to genetic testing, which of course means there's inequitable access to diagnosis, whatever. Inequitable access to genetic counselors, the specialist. Even once you have a diagnosis and I have XYZ, can I get to a specialist for XYZ? And even if I can get to a specialist for XYZ, am I in a multi-specialist center who can deal with all the different organ systems that are impacted? Or am I just dealing with, say, a pulmonologist, or just dealing with a neurologist? Do I have access to psychosocial, financial, or educational support for families? And I'll tell you the answer is frequently no. And then, do I have access to clinical trials or other types of research? For instance, natural history studies. Natural history studies for rare diseases are incredibly important, as you'll hear later in the talk. But not everybody has an opportunity to participate in these. And even for the most motivated patient or parent of a patient, because frequently rare diseases manifest in children, participating in clinical trials is often not an option. For the same reason, participating in clinical trials is often not an option for, say, patients with cancer or other things. Just, the difficulty of getting to a trial site, difficulty of taking time off from work, the need to have a caretaker attend with you. The lack of being invited to participate in a trial, the lack of understanding about what research is, et cetera, et cetera. And then, of course, I'm talking about all these list of things that, some patients have easy access to, many patients don't. And at the bottom of that is in a few interventions. In a few interventions may be of enormous interest to people who know that that's the only way they could possibly have a treatment for their condition or their child's condition. Very, very few people are going to get access to this for reasons that we talk about. So now let's go back to the rare diseases world for a moment. We're out of individual for a second, rare diseases, and just to sort of situate what's happening there. This is the most recent gene therapy that was just approved by the FDA. I want to say like last week, but maybe it was 2 weeks ago or something. Lenmeldy for metachromatic leukodystrophy. So this is the first treatment for MLD, which is otherwise fatal. So, the community is super happy. Not so happy about the price tag, $4.25 million for a one-time treatment. But, in terms of cost benefit, it may be worth it. This is a drug that, this is a gene therapy that seems pretty much curative, although cure is a loaded word. And how do we know it's curative when we don't have long-term data, et cetera, et cetera? But children who would have definitely died otherwise are now thriving, seeming pretty much normal if they receive the treatment before symptoms appear, or in some cases, right after the first signs of disease. But, this is a rare disease. Estimated in the United States, about 1 in every 40,000 US newborns. Companies can't really live on that size of a population, even if they are charging 4.2 million apiece. So the company is working to expand newborn screening efforts, both in the US and Europe, to find more patients. And this is because A, the drug's not effective unless it's given at this precise point in time early on, and B, they need patients, a pool of purchasers in order to stay viable. So that's one gene therapy that I want you to keep in your head. Again, not an individualized therapy, just a rare therapy. And then here's another one. I don't know actually how to say this. I'm going to say Glybera. If someone knows that that's not how to say it, maybe Dr. Bradbury, you can correct me. But this was the first gene therapy to win regulatory approval in Europe. And they found out the hard way that if you don't have patients, you don't have a viable drug. This was withdrawn due to lack of demand. It was commercially unsuccessful. So, we see these prices and go $4.25 million. That's unconscionable. No one should have to pay that to keep their child alive. I agree. I feel sort of sick when I see those price tags too. But when you're dealing with a very small population, somehow you're going to have to make this viable for a company to continue, because companies are for-profit entities, unless you're going to take the for-profit out and have this be something that is, done by a philanthropic foundation, a government, etc. So that's a question I leave for you to ponder. So here's our picture back again. And as I mentioned earlier, you can tell that something like MLD, which deals with a rare patient population and needs to be given at a very precise moment in time, is not going to work for a developmental pathway like this. And indeed, you can see this is from the press release from the company, Orchard Therapeutics. The FDA approval of Lenmeldy is based on data from 37 pediatric patients. Okay, so in this thing that I put up here, you can see we had upwards of 3,000 volunteers for this particular drug coming to market. Rare diseases, we're talking much smaller subject pools. So 37 pediatric patients. And the patients were enrolled in 2 single-arm open label clinical studies or expanded access. So when I said earlier in phase 3, you might have comparator trials where you are randomized to a placebo or getting some other drug. Well, in this case, there was no other drug. This is the first drug for this disease. And it was deemed not appropriate to randomize to a placebo because in this case, they had the good, rich natural history data. They knew that these children were going to die. They knew sort of the time frame in which they were going to die, et cetera, and said, we can just collect data from these treated children, whether they're in a trial or whether they're getting it outside of a trial in expanded access, what a lot of people know through compassionate use, and compare that to the known course of the disease. And the evidence was convincing enough in these 37 patients that we are comfortable approving the drug as a therapeutic to be marketed. So the point I'm trying to make is when it comes to, regular drug development and then drug development for rare diseases, we've got some differences going on. And those differences can be the size of the trials, the type of the trials, and most importantly, what type and amount of evidence regulators, not just the FDA, but regulators around the world deem sufficient for regulatory approval. And remember, I said earlier, my question is why? Why do we differentiate between these levels? Is it just a pragmatic thing of saying we're never going to get 3,000 plus patients for something, in MLD? So we have to differentiate for pragmatic reasons, or is there something more going on here? Why can't I go to the next page? There we go. And so what I want to drive home is when it comes to regulatory approval, it's this hurdle, right, that companies are trying to get over with their packets of data and information that they're giving. But this hurdle is not something, that is natural law or handed down by some deity that we must follow. These, these levels of evidence that are required are something that are set by the regulator, and they're set by the regulator and influenced by feedback from us, the general public, patient advocacy groups, sometimes legislators who get involved. So these hurdles that you see, they can go up, they can go down, and they don't have to go up or down in lockstep across all disease areas. In certain disease areas, one can go down while the others stay level. Or as we saw in COVID-19, at certain times, when we're in a pandemic and everyone's worried about, oh, we need drugs fast, the levels can go down, not necessarily because COVID was a terribly fatal disease. It was not a terribly fatal disease, but because there was fear about the disease and concern about its economic impact, et cetera, et cetera. So there are things that can affect what level of evidence we expect and why, and these things are socially mediated. So that's the point that I wanted to make. And this gets back into the regulatory flexibility. So when you have someone from, say, the ALS community saying, look, the drug does not seem to work well, but it works a little bit, and that's all we're asking for. We have a fatal disease, and we will take a little bit. Why don't you use your regulatory flexibility? And instead of looking for a, P, 0.05, I'm not a statistician, take something that's off just a little bit, because that little bit has meaning to us. This is what people are talking about when it comes to regulatory flexibility. All right, so that is what we're talking about with rare diseases. People saying, shift the goalpost a little bit, be flexible in your interpretation, understand you can't hold these rare diseases to the same metrics that you would hold a more common disease. Now we're going to move down to individualized therapies, where obviously the same argument would hold true. If you're designing a drug for one person, obviously you cannot expect the same volume of data to be given to the FDA. But more importantly, why would you even be trying to bring that drug to market if there is no market? So this is a case example. This is Alison, and Dr. Bradbury is going to talk more about her. So I'm just going to hit some key points dealing with sort of the regulatory aspects of her case. So in 2018, the first individualized antisense oligonucleotide, or ASO, was created for Mila, who is the little girl that you can see in the picture. She had a previously unknown variant of Batten's disease, and I'll skip some of her medical history here because Dr. Bradbury will deal with it. But through the wonder of the internet, her family was put in touch with researchers at Boston Children's Hospital who ended up repurposing a approved existing ASO treatment into something that was bespoke for her form of Batten's disease and ended up making the world's first N-of-1 treatment. And the time from diagnosis to first dosing, 1 year. Think back to that slide I keep showing you of, multiple years. 1 year, 1 year is unheard of. The treatment was associated with a reduction in Mila's seizures, so her family was happy about that outcome. But in the second year, her improvement plateaued. The family ended up withdrawing her from the drug, and Mila died in February 2021. Now, Millicent was estimated to have cost in the ballpark of 3 to 5 million. Now, that's a very large ballpark. I don't know why it's such a large range, but that's a lot of money. And especially for someone with, say, a public health background, you're like, wow, we could have done so many other things with that money. But it was money that was fundraised by private foundations, including one that was set up and run by Mila's mom. Research grants, because, research grants are there to pursue cutting edge research, and the hospital. So it's not like, we were taking money away from, a public health program or taking money away from other things and redirecting it to Mila. This is money that was raised for the purpose of trying to help Mila. People typically are very generous when they think they're rescuing someone from dire circumstances. And it was used to create this drug for Mila. Now, obviously, this creates access and equity concerns. You can't do this for every person who has a unique variant of some disease. Even if you were to rely on the generosity of people who are willing to give charitably, the interest just burns out after a while. How many campaigns would people be willing to respond to? And we know from the crowdfunding literature that there is a type of appeal that people respond to. Cute kids that have a family unit that is social media savvy, et cetera. And there are people that are just not going to be able to get money with that formula. So this obviously has access and equity concerns. But the concern I want to focus on right now is, again, my question of why do we treat these things differently? So Janet Woodcock and Peter Marks, who are both longtime FDA officials, actually wrote an editorial at the same time that Mila's N-of-1 therapeutic was printed in, I think it was New England Journal. And I'm not going to read this to you, but the point is that right from the get-go, they were raising this questions of, in N-of-1 circumstances, we can't hold it to the same level of evidence we have done before. We can't demand a phase 1, a phase 2, a phase 3 trials because there aren't people. Especially if we're dealing with someone who's rapidly progressing with a fatal illness, we can't even necessarily say they need to do all of the preclinical and animal work that we would have demanded in a clinical trial because the person will be dead before that happens. So how much should we demand in these circumstances versus how much should we just be like, just let them try whatever they want to try? Because if we throw our hands up and say, let them try whatever they want to try, that's basically, just the regulatory state stepping back and saying, we have no role here. And then also, what happens when another Mila shows up, someone who has that same variant of Batten's disease, and suddenly it's not an N-of-1, it's an N-of-2. or an N of 3. And suddenly we said it was okay because it was just one person risking their life, but now it's multiple people. Do we say, well, stop now, we have to do it as a trial, or how does this work? And these are the questions that you can see are actually coming true if you look and see what's happening after the Mealison case. So far, there's been no other patient identified with Mealis mutation. There was another case afterward, a drug called Valericin, because the first child to get it was Valeria. In this case, it was 2 children. And, it's a question of how do we handle this when it's 2 children, not just 1. And then there was JC Fusen, because the first recipient was JC. And in this case, the FDA allowed a small number, I don't know what the number is, maybe 5 or 7 or somewhere in that, to be treated with JC Fusen. But this was a genetic form of ALS that we knew there was more than 5. We knew there was more than 7. We knew there were a number of these patients out there. So FDA at some point said, enough. If you're going to treat any more people with this JC Fusen, it has to be done as a trial. And again, the question is, well, you already waived all these requirements for these other people. What's the rationale for now going back and saying, no, we're not doing this for anymore? So, as I said, I'm raising these philosophical questions, and I'm not a philosopher, so we'll throw these to Dr. Howard later. And then the question is, where is this line, right? So in the FDA guidance, it again, talking about ASOs, it's really talking about typically 1 or 2. In Lorem, the organization that I talked about earlier that is creating these individualized ASOs charitably for people in need, sets the cutoff at 30. I mean, 30 is quite different from 1 or 2. And then, there's just the obvious point of you may think that there's only 1 or 2 people in the world with this unique genetic mutation, but once you start looking at it, you're going to find more. And the whole reason that we've singled out these things as saying we're going to treat them differently, we're not going to make them go to trial, et cetera, is not only because they're, so incredibly rare. We do make incredibly rare, ultra rare things go through trials. I just showed you that with the MLD. It's not only because they're so rare, it's because we think that they're not going to be commercialized. We think that they're so rare that no for-profit company is going to be willing to take them on, bring them to market, and then try to make them available. And that, to me, does not seem like an incredibly compelling reason to decide how much evidence we should demand of something, just whether if there's a commercial capability or not. But again, I'm not a philosopher. So I will just close with saying, to me, it's a question of how do we justify that one patient or patient group with a dire prognosis must go through this traditional clinical trial process in order to try a novel therapeutic, when another with an equally dire prognosis, but a known prevalence small enough that we predict no commercial interest is going to result, can be permitted to try a novel therapeutic after significantly less safety testing, et cetera. And I just think to myself, the person who is dying from cancer, or the person who is dying from TB or whatever, who says, I am desperate. I understand it's a long shot. This is my informed voluntary consent that I'm willing to take on the risk because I have no other option. And we say, well, we're sorry, there is a trial and you would need to go into the trial, and the trial is only at phase 1 or phase 2, and it's in Nebraska. So you're going to have to go to Nebraska. Like there's a routine here that you would need to follow. And only if you don't qualify for the trial, you can petition the company making the drug to try to get it through compassionate use, because that's how our system is set up. Whereas someone who has an equally dire prognosis, but there's just no likelihood that any company is ever going to be interested in commercializing the therapeutic, we say, okay, we'll let you and your doctor do this as a sort of, innovative treatment. And that's what this slide says, so you don't actually have to read it. But it was just saying, like, our social contract when it came to clinical trials was you don't get to opt out of them. You don't get to say, no, I want to try this new product, but I'm going to opt out of the clinical trial. Thanks very much. Because clinical trials is how we get information about new drugs, how they come to market. The data gained is a social good. And we know people would opt out of clinical trials if they didn't have to participate in them. So we set up the system where if you are able to participate in clinical trial, you meet the inclusion criteria, you're physically able to make it to the trial site, et cetera. If it's feasible, we're not going to say like you have to fly to London, but if it's feasible, then in order to get the novel therapeutic that you want, you have to participate in the clinical trial. Now, if it's not feasible, we do have this sort of, safety net of expanded access. But for the most part, we want you to go into clinical trials, except for now in this individualized therapeutic world where we say, well, we've decided for pragmatic reasons and commercial reasons that we're not going to have a trial and we are going to hold everyone to this lower bar. That may be appropriate. I'm not saying it's not appropriate. I'm just saying it's something I think we need to put more thought into about why we're doing that. And this gets into this question about, are we giving these products to people as clinical research with this goal of learning generalizable knowledge that will be applied to other drugs, other diseases, et cetera? Or are we giving this to people saying, yeah, it's experimental, but our main intention here is to help you. So this is clinical care. And so in conclusion, when it gets to these individualized interventions, we've got regulatory challenges, we've got informed consent challenges, and IRB has to juggle with risk and benefits when both of these are pretty unknown, funding challenges, equity challenges, both with regard to individuals getting access, as well as, trying to treat different conditions fairly. And then, of course, they also are this new era of medicine that offer treatment opportunities that we've never had before and can actually, really help people who we've never helped before, or at least that's the goal. That's the hope. We haven't seen it fulfilled yet, but that's the hope. So I will stop there and turn things over to Dr. Bradbury for her to answer some of those questions, and then Dr. Howard can answer all the rest. So thank you for your attention, and I am taking my slide down now, I think. There we go.
Thank you very much, Allison. Dana, can you see the slides okay? All right. So I would like to thank Dana for allowing us to have this, what I think is a very important discussion today on the ethics of rare disease and individualized therapeutics. So I wanted to just start off real quick with the landscape for rare disease therapy development when this new field of individualized therapy kind of came to light more recently. So in 2016, the FDA approved an antisense oligonucleotide for SMA, or spinal muscular atrophy. And so this drug went through the pipeline that Dr. Bateman-House just so eloquently detailed, where they enrolled as many patients as they could in a set clinical trial site. They were able to prove safety and efficacy of this drug. And because they were able to bring this drug to market approval, now over 14,000 individuals with SMA have been able to receive this market approved drug to date. So this is a rare disease, but a drug that has taken the kind of typical pathway that we think of and is now market approved. So just 1 year after the FDA approved the ASO for SMA, that's when Millicin came to light as the first individualized therapy. As Dr. Bateman-House already mentioned, this was an ASO that was developed in Boston for this one child, Mila. And this was published in the New England Journal of Medicine. It also quickly garnered a lot of attention in the press, including this article in the New York Times, where they really rapidly started grappling with some of these questions about, is ultra-personalized medicine fair, and how it's delivered and in its regulation. And I wanted to kind of highlight a little bit more on the science side, the expedited time frame of this particular drug development. So Mila was diagnosed here in November of 2016, and the exact mutation was identified in the spring of 2017. About a month later, the patient cell lines were established. And then in the fall of that year, 2017, an ASO hit was identified. About a month later, they were able to demonstrate functionality of that ASO and started toxicology study in rats of December of 2017. Just 1 month into the tox rat study, they received permission to start dosing the patient, and the patient was dosed in January of 2018. So in opposition to the over decade of drug development for the ASO for SMA, this was really rapid. It was about a year, but really 10 months once the actual research was ongoing, and keeping in mind that the toxicology study was only 1 month in when they allowed this patient to be dosed. So this was really an unprecedented time frame of allowing a drug to reach a patient. Just 1 year later, in 2018, was the next individualized antisense oligonucleotide. Again, Dr. Bateman-House mentioned this. It was for a devastating form of epilepsy. And the initial patient, the family raised more than $1 million, and they started a foundation. And they even moved to Massachusetts to be close to Dr. Yu to be able to help support the efforts to make the antisense drug for their daughter. And in 2020, the drug injection began. This is an intrathecal delivery that has to occur repeatedly. Unfortunately, this first patient developed hydrocephalus and ended up resulting in her death. A second patient that had the same disease, Lucy, was also given the same antisense oligonucleotide. So again, this quickly became from an N of 1 to an N of 2 for the same drug product. And Lucy also suffered from hydrocephalus. And this father really, brings up this really, important point that I think families really struggle with, which is that, they have this hope to improve the quality and the length of their child's life, but they also know that they could make their life shorter. And this is just a really, really difficult decision for families to have to make. And one thing with these individualized therapies is that there's less of an incentive to share the data and to publish the work. So although this happened from 2018 to 2021, there still hasn't been a publication detailing these antisense oligonucleotides and the cause of the hydrocephalus. Therefore, the research field as a whole hasn't been able to learn from this and to help to advance the field safely. As Alison also mentioned, there's been initiatives surrounding these individualized medicines. One of them is the formation of INLORM, which is this foundation that is developing ASOs for nano-rare patients. So as Alison mentioned, 30 or fewer patients. To date, they've screened over 225 applications, and they've made 6 ASOs for patients. There's also this N of 1 initiative that was started by Dr. Yu, and they're putting together work groups to really focus on different aspects of individualized therapy, including how to identify the best patients, what's the most appropriate preclinical design, how do you coordinate data, also clinical outcome measurements, and clinical implementation of these. So there are some new initiatives surrounding this individualized therapy development. I didn't want to only focus on ASOs though, because there's other genetic therapies that are following suit. So in 2019, the FDA approved an AAV, or adeno-associated virus gene therapy, again for spinal muscular atrophy, or SMA. This drug, again, went through that traditional pipeline of phase 1, 2, 3 clinical trials where they enrolled as many patients as they possibly could in clinical trial sites. they were able to demonstrate both safety and efficacy of this one-time delivery of an AAV-mediated gene replacement therapy. And now because of this and the market approval, over 3,000 kids with SMA have received this gene therapy. However, there's other ways that gene therapy has been delivered to patients, and Alison mentioned this expanded access. Also, it used to be referred to as compassionate use. So this was one gene therapy clinical trial in 2019, so just after SMA's approval, for a disease called Tay-Sachs disease. And the first 2 patients here that were published in Nature Medicine actually received the gene therapy in an expanded access trial. A regular clinical trial did start after that and has enrolled a number of patients since then. But I think this brings to light, some questions about, is expanded access justified if a trial hasn't started yet? So is there drug product, but the trial isn't underway? Can you go ahead and dose some of those patients that may not make it to the clinical trial start date? But then what if the clinical trial has started and there's patients that do not meet the inclusion-exclusion criteria? Is expanded access still justified for those patients? And for a lot of these gene therapy trials that have an industry sponsor, this has become really complicated because they really want to try to get their drug to market approval. But if they take drug product away and give it to expanded access patients, it can kind of muddy the water of what their clinical readouts are looking like. And it's put some of these, companies in a bit of a dilemma with how to handle some of these patients that need really immediate attention. And very different from expanded access, in 2022, there was actually the first individualized AAV gene therapy developed. This Canadian family raised over $3.5 million to develop an AAV gene therapy for their son's rare condition. And this was published in August of 2022. And they said that the family was optimistic 3 months after the child received the gene therapy. But again, this father, really struggled with the dichotomy that this was really exciting. But the last thing they ever want to do is to hurt their child. So these families, have this real hope in trying to find a way to help their kid's disease, but it's also very risky when there's no prior data in humans. So I just really briefly wanted to touch on the landscape for researchers. So, we kind of get asked, why are you working on these diseases and what has changed to really make this, really an uptick in research in recent years. And one of them is advances in allowing for patients to be identified. So now with whole genome and whole exome sequencing, we're able to identify disease-causing variants much quicker. And also the functional assays to confirm the mechanism of disease are happening at a much quicker rate. And some of these companies, along with giving the diagnosis, so GeneDx, for example, will do a targeted RNA study and help identify splice site defects and what's happening with those variants. There's also a lot more resources to educate patients and families. There's annual meetings. These patients now are coming armed with so much information about how these prior ASOs and AAVs have made it to kids. And so they're a lot more knowledgeable. So just some quick examples, in the lab, we'll have some patients that, in our own Institute of Genomic Medicine can get diagnosed with a rare disease. On one of their clinic visits, they can give us blood samples. We can now quickly take their PBMCs and reprogram them and make human-induced pluripotent stem cells. From these, we can make specific cell types like neurons or even develop three-dimensional organoids, which can recapitulate some of the disease phenotypes. And we can do single cell RNA sequencing, figure out what's going on with this disease, and then use these in vitro systems to screen different therapeutics. And this can all happen in a matter of months. We can also use gene editing to make in vivo models. So we can mimic a specific patient mutation. We can generate a mouse model in under a year, and then we can do clinically relevant readouts, such as we can track seizures and see if different therapeutics are able to correct those diseases. We also have a growing molecular toolbox with how we can treat these diseases. So we'll have these in vitro or in vivo model systems, and we can study the molecular changes and how that impacts protein structure and function. And then, for example, if we determine a loss of function disease that has a shortened or degraded protein, we could use a gene addition therapeutic strategy. could modulate them with antisense oligonucleotides. Or in contrast, if there's a gain of function, we can use knockdown strategies. Again, you can use antisense oligonucleotides, or we can use gene correction with some newer gene editing techniques such as CRISPR gene editing or prime editing or base editing. So we have this ability to create these patient-specific in vitro and in vivo models, and then we have all these tools that we can then try to really strategize the best way to tackle these different diseases. However, I think recently we've had some lessons learned about individualized therapy. So this was, a pretty well-known story in 2022, where the first patient to receive an AAV-mediated CRISPR gene editing for muscular dystrophy passed away. So the patient went into cardiac arrest 6 days after the treatment and died 2 days later. And this was a bit of an interesting situation because it was the patient's older brother who helped to develop the foundation, a nonprofit foundation, Cure Rare Disease, that supported the development of this therapy. And lastly, I want to touch on the landscape of accessibility in rare disease space. So we can try to start to kind of identify what some of the barriers are in accessing these therapies. And really, for the market-approved therapies, each of these rare disease gene therapies has been competing for the world's most expensive drug. So in 2018, the first AAV gene therapy, Luxturna for eye disease, had the US's most expensive drug at $850,000. However, in 2019, 1 year later, this was overtaken by a $2.1 million price tag for the gene therapy for spinal muscular atrophy. And then this price tag was increased to $3.2 million for the Duchenne muscular dystrophy and $3.5 million for hemophilia gene therapy. And these were in 2022 and 2023. And then, as Alison mentioned just a couple of weeks ago, the gene therapy for MLD came on the market, and now this is the world's most expensive drug at $4.25 million. And I really love this piece that was by Glenn Pierce, who's the vice president of the World Federation for Hemophilia. And he discusses that in the US, the treatment for an adult with hemophilia averages $700,000 to $800,000 per year. So the price of this gene therapy will pay for itself in a relatively short time. But patients in low and middle income countries, they don't even have the access to the treatments that we have in the US that cost the $700,000 to $800,000 a year. So those are the patients that would benefit the most from these technologies. However, they're the ones that can least afford to pay, and we cannot leave the majority of the world behind. So this is my email, and I know that we want to just open it up to questions by Dana and the audience. So I'll stop there and happy to answer your questions now. Dana, you're on mute.
Sorry, I was saying brilliant things. I was just thanking Dr. Bateman-House and Dr. Bradbury for just giving us this real, important overview of the staggering sort of, the staggering sort of speed, velocity in which these things are developing, and really bringing up some really difficult ethical questions. We went a little bit over in the presentation, so I'm going to jump into the Q&A from the audience. So feel free, audience members, if you have other questions, to start asking them. And then I'm going to return to some of the questions, but you covered so many of the ethical questions that I was already thinking about. So the first question, I think, is a great one from Gregory Musselman, who is it's a big picture question, but I mean, one of the things that really is highlighted in both of your presentations is how one of the interesting things about this is that it's no longer business as usual when it comes to really thinking about research, drug development, incentives, and also then approval. It's just like completely different, right? So why would a company, so this is Gregory's question, why would a company even want to sell a product if there's such a small consumer pool? Why not give it to a governmental agency and have it be sort of either governmental agency responsibility I think as a political philosopher, there's some ethical issues related to that. So how should we think about, the commercialization of these drugs, and why are people doing it, and how can this be a sustainable research endeavor? Maybe we'll start with Dr. Bradbury, and then we'll go to Dr. Bateman-House.
I think it's a great question. And unfortunately, in the gene therapy space, especially for rare diseases in recent years, we've seen a number of companies try to make this work and license intellectual property and take a AAV drug product and really try to, make this a product that they can sustain, but in a lot of cases, this just hasn't held true. So there's been a really unprecedented number of gene therapy products that have been returned to academic institutions in the past couple of years. And this has been, very difficult because we think a product is, going, to be supported by a company, and then it ends up back in our hands. And this can be obviously really frustrating for the families too. So there's obviously some success stories, like we know for spinal muscular atrophy, but then there's been a lot of programs that unfortunately, this just, the business decisions just haven't been sustainable. So I think it has to look at the bigger portfolio of the company as a whole. And maybe, if it's some of them balance some of the rare diseases with not rare diseases. And so they're able to kind of take these rare diseases, under the wing and make it feasible. And then sometimes, unfortunately, it's just not so. But I think relying on the government to provide all of these really expensive gene therapies, there's just not the structure right now, in our government. There is no way to provide all of these rare disease gene therapies supported by our government. So if there was, some outlet to do that, but they struggle to pay for the ones that are already developed.
Right. Dr. Bateman-House?
I would just say, if you look across the landscape of particularly gene therapy, you can see companies are increasingly moving towards common diseases, heart disease, stuff like that. So I think some of this was starting with rare diseases because a monogenetic disease was easier to get your proof of concept. And now people are moving into your more complex, but also more profitable diseases, which of course is unfortunate for rare disease communities.
Mm-hmm.
I think a number of companies were taking advantage of the fact that if you got an approved drug or gene therapy in a pediatric population and you were the first to do so for that indication, you could get a pediatric, I just forgot what they're called. There were very large financial incentives for bringing new drugs to market for pediatric populations, that even if you didn't make money bringing the drug to market, you could make money by getting that pediatric incentive, which you could then turn around and sell to other companies that they would be able to use to get a priority review from FDA. So there's like this very complex business mechanisms going on.
Thank you. And one further sort of question related to the political, philosophy angle of it, of, having governmental organizations, as Dr. Bateman-House was bringing up. A lot of these research endeavors are being done currently through philanthropic organizations that are geared to getting more money for this specific endeavor. But then once we start thinking about the NIH or some other HHS paying for these very rare diseases, it's no longer this sort of extra pool of money, but whatever the pool of money that is already there.
Can I just say, I've talked to numerous patient advocacy organizations that once a for-profit company has surrendered the patent or the IPO back to academia, are interested in licensing that intellectual property and setting up shop themselves, saying, if nobody else is going to make this drug, we are going to make the drug for our community. which on one hand is great. On the other hand, you get into the sticky wicket we have seen with the cystic fibrosis community, which has actually done this in terms of if you have a very, very, very expensive product, people start saying, well, wait a minute, Cystic Fibrosis Foundation, are you a for-profit drug manufacturer or are you our advocacy organization? Because if you're our advocacy organization, why are you charging us so much for this drug that we need to stay alive? So
yeah, so maybe this second question is related to some of these issues that you just were talking about. So I'm going to skip around, but this one is from Skylar Martin. So Skylar Martin says, a big part of tapping into those limited patient populations involves this proof of concept, broadcasting awareness of a new therapy, new therapeutic, both for providers and for patients. Are there unique ethical considerations to the sales marketing efforts that pharma might be taking in this space that is different from other existing sales and marketing efforts in general medicine? And I just want to add one more sort of question that came, I think is an interesting one that you've been sort of touching upon, but I would love to just hear your thoughts about what is the distinctive ethical issues that come with researching the rare disease community. You've been mentioning some of the things, the way the internet finds people together. So can you tell us a little bit more about the ethical issues that come up with this very specific and sometimes very knowledgeable population of patients and families? Maybe we'll start with Dr. Bateman-House and then we'll go to Dr. Bradbury.
Dr. Bradbury is probably going to be better since she's the researcher, but so I will say, as you mentioned, many of these rare disease communities are very small, are very tight knit, and I would say probably more so the smaller the community is. Although all communities are going to have their portions that are left out. So, you know, there's equity and access issues to knowledge even within the smallest community, right? Because if a lot of this conversation is happening online, not everybody's online, not everybody's English fluent, et cetera. But so there's a lot of concern, especially if you're trying to do a blinded trial or whatnot, with people sharing their experiences online, saying, you know, I got the infusion today and I had no response whatsoever, so I know I'm on the placebo. Or, you know, I had an infusion today and within 24 hours I had a high fever. So if you have a high fever, you can presume that you're not getting the placebo. So a lot of concerns about, you know, are you potentially, you know, interrupting the sort of scientific veracity of the poll by having people sharing these sort of anecdotes outside of the confines of the actual clinical trial. There is a huge overlap between sort of the patient advocacy personnel within pharmaceutical companies and actual patient advocacy groups. There are co-sponsored meetings, there's co-sponsored educational material, there's co-sponsored internet forums, et cetera. And not always is that stuff branded. So there's not necessarily the transparency you'd want to know that this is a company X funded forum or things like that. And then last but not least, I mentioned earlier the importance of natural history data. Many of these natural history data are in registries that the patient groups themselves have run, or perhaps, you know, an academic researcher who was doing it with funding from the patient advocacy group. And that data is of huge importance to the pharmaceutical company. But there's questions about who owns the data, under what circumstances can it be shared? You know, what happens if you have two different registries and they didn't use the same terminology? How do you integrate that data? So there is a huge push right now for data integration. But I think you also need to figure out how do you do the data sharing, the confidentiality, the ownership, et cetera.
Yeah, I'll just kind of go on that a little bit more about the inequity of access, because these are not commercially viable, at least the vast majority, it is relying heavily on families and foundations to raise the funds to support the development, the manufacturing, and then the clinical implementation of these. So sometimes this can have, what some people call like pay to play, which, it's only those families that were able to support it that have the ability to receive this treatment. And also at another level, if there needs to be, for example, how I showed in vitro or in vivo model systems, the patient that was able to donate the blood or the skin sample or the mutation that you select to make the mouse model, that's the development of the individualized therapeutic is driven by a patient's genetic makeup and the motivation to treat that one patient or a handful of patients. So again, it's an inequity of access based on who is able, to participate in the research. And then another thing that I think I'll point out is the really what becomes most contentious is the time that it takes for these, because most of these are pediatric indications. A lot of them are rapidly progressive. So the patients are, time is really critical for some of these patients, the time to intervene. And there's an urgency. Patients might be, losing developmental milestones. They might be acquiring seizures or becoming resistant to seizure medications. And every week or month is really critical. So there's this urgency to get a therapy. However, performing rigorous and robust scientific experiments takes time. So I think that's, one of the difficulties specifically with rare diseases is how to move things at an appropriate pace, while we know a lot of these diseases can be really progressive.
Can I just add one more thing?
Mm-hmm.
So another challenge with rare disease is frequently you end up having to recruit across national boundaries just because you need the numbers. And I was speaking with one company recently. They are recruiting, I think, in 6 countries, and showed me something on the order of like 40 different consent forms, because each country had their own regulatory requirements of what the informed consent had to have. And then they needed to translate into different languages. And, then there was some pediatric consent forms, some adult consent forms, and just, the time it takes when you're trying to navigate that sort of stuff. As Dr. Bradbury was saying, if time is critical and you want to do things right, you want to follow the rules, you want to make sure people are getting right information, but some of that just takes time.
And it seems like with the recruitment going cross borders, there's going to also be the worry of the patients that are being recruited right now for research participation. Patients like them in the future may not have access to the therapeutics once they're developed.
Well, and also FDA for certain types of gene therapy is asking for at least up to 15 years follow-up.
Mm-hmm.
And the question is, how do you do that? Companies go out of business or they merge or what have you. And so, what happens if you're on year 5 and the company goes out of business? As Dr. Bradbury said, we want that data. Like, we need to know what happens long term when people get this, transgene and it integrates into them, and we want to know what happens. But, is it the government's responsibility at that point to pay for the follow-up, or who?
Mm-hmm. So one question that is an interesting one that just came up from an anonymous attendee is whether or not the FDA is doing this backwards. So so the question is, should the FDA view the risk, think about hastening approval of certain drugs for rare diseases in the same lens as other more common diseases, since they affect more people? So basically, what I take the question to be is this sort of lowering the bar for evidentiary standards, as Dr. Bateman-House was saying, actually incentivizing the sort of development of rare disease research and maybe the FDA should take a different approach and focus more on like approving or diseases that will affect a higher population.
Yeah, I mean, I don't want to get into the evidentiary bar part of it, but you could certainly say we've had great success directing companies into rare disease. We've had less success, but still some success directing companies into pediatric disease with these financial incentives. Couldn't we do the same with, you know, neglected tropical disease or, you know, some of the most common killers? I mean, that makes sense to me as someone with a public health background, but I'm not the one running the show. I guess I will also say, you know, those incentives are tied to being first in market. So if it's not first in market to say coronary heart disease, you wouldn't get these incentives. But there's no reason why you couldn't come up with different incentives, I would imagine, says the non-health economist person.
I would say the, you know, approval for all drugs sometimes seems, you know, painfully slow. So finding ways to improve, you know, the pathway for common diseases and rare diseases, I think they can help each other. But I do think there were some interesting learnings from the COVID pandemic and how suddenly drugs were rolled out in a much faster pace than had ever been before. So one thing that the FDA is actually piloting right now is enhanced communication between the FDA and sponsors to see if kind of more frequent back and forth between the two can help expedite the process of getting drugs to approval, because they found that when the pandemic happened, you know, there was this constant back and forth and checking in and looking at data at different increments, and that maybe that was helpful. So they're actually piloting that right now. So I'm interested to see if the enhanced communication touch points between sponsors and FDA will help some of expedite some of these processes.
I'll go out on a limb and editorialize one thing. You know, we have the accelerated approval pathway, which when they say for indications of severe unmet need, you can bring a product to market based on less evidence around the phase 2 part of drug development. And then the company can sell the drug, it can do whatever it wants to do, but it has to do the phase 3 trial, even though it's already on the market. And then the results of the phase 3 trial, if they're positive, great, nothing happens. If they're negative, FDA has the prerogative to pull the drug off the market. And we've seen this happen recently with a drug for ALS and one for Alzheimer's and whatever. FDA actually did this for a gene therapy drug recently. And I actually think that was a bad call, because unlike small molecule drugs, where if you find out it doesn't work, you can just discontinue the drug and, you know, in a matter of hours, days, weeks, whatever, the drug will wash out of your body. You can't do that with a gene therapy. And especially given that the phase 3 trial was almost done at the time that they granted the accelerated approval, I thought, why is FDA saying go ahead and let these people get this irreversible product when if we wait like, you know, 3 more months or something, we're going to have the phase 3 data. And I completely agree with Dr. Bradbury, you know, for these life-limiting, progressive, severe diseases, time is of the essence. If you miss your window, you miss your window. But at the same time, I was just like, it's irreversible, you know, we don't know its duration, we don't know all of its side effects. I think I'd err on the side of prudence for this one. So
yeah, so we have only 1 minute left. And so I want to give both of you sort of one final thing to put on the table that we haven't quite mentioned yet. And one thing that I want to sort of highlight, this was more of a comment than a question, but I think it's an important perspective to keep in mind. So this is from one of the audience members. Why is health so closely related to cost? drugs for not so rare diseases still cost too much. I have MS and my drugs cost about $50,000 a year. Sickle cell is prevalent in my family, and the new stem cell transfusions for sickle cell are not feasible. The audience member says we need government company partnerships. One of the things that has been talked about a lot is sort of concerns about justice and equity and access. And I really appreciate, Dr. Bateman-House, how you were bringing this up, all of this conversation in the context of already unjust, sort of inequitable distribution of health resources. So what final things should we be thinking about when it comes to the sustainability and sort of developing this into a kind of therapeutic model that will be beneficial to the most people in the future? Maybe Dr. Bateman-House first, and then Dr. Bradbury, you can have the last word.
Well, someone just mentioned cost of medicine, so I guess I'll go here with my last comment. What we have found in the last few years is that if payers, insurance companies, national health services, or whatever, feel that the FDA evidentiary bar has become unreliable, they become the gatekeepers. They will refuse to pay for something. So the question is, do we want to have one gatekeeper and one arbiter of, this has been proven or not, or do we want to have numerous gatekeepers and arbiters, which then makes equity and access, more of a scattershot thing. So
Thank you.
So one thing that I think has been helpful in the last few years is the NIH National Institute of Health has put out new programs such as the Urgent, which is ultra rare gene therapy initiative, and then the Bespoke Gene Therapy Consortium. So I think some government resources are finally getting behind us to help us actually run these preclinical and translational studies. The next thing that I would say is for the FDA to start considering these as like a platform technology. And once one is approved, how do we take that approval from one and apply it to another without reinventing the whole wheel? So thinking of these as platform technologies. And lastly, I think to really make these globally accessible, we need to rethink manufacturing. We're not going to be able to take manufacturing at the scale that we are and make gene therapies that we can then ship out to, every country to make sure that kids around the world have access to these. So finding a way to scale the manufacturing at an affordable rate to make these therapies really global.
Thank you, Dr. Bradbury, Dr. Bateman-House, and thank you, audience members, for the amazing questions. This is clearly a developing conversation, and so maybe in a few years we'll have another panel, and we'll see where we are. And yeah, have a good afternoon, everyone.
Thank you, Dana.
Thank you.